Hi, everyone. So, today we're going to talk about mainly epithelial and connective tissues. I do want you to know that the four primary tissue classes in this lecture are epithelial, connective, muscular, and nervous. I'll have more for nervous tissues and muscular tissues later in the semester when we get to the nervous system and the muscular system. Bone too. Once we get to skeletal system, we'll look at some bone tissues, but I want you to know that the four primary tissue classes are epithelial, connective, muscular, and nervous. This lecture will cover a lot of epithelial and connective tissues. At the end of the lecture, we're going to look at some slides, and there are going to be some muscle slides there, but that's just so you can get accustomed to what they look like. And there's going to be one nerve cell, but we're going to look at muscular and nerve more when we get to those chapters. Right now, we're going to focus on epithelial and connective tissues. So, prepping tissues. We look at histology. Histology is the study of tissues. We look at it at the microscopic level. So we have to prep those tissues to view them through microscopes. Here we have three different examples. This first example is a fixed slide right here. So it's been thinly sliced, right? It's been sectioned, it's been stained, and it's ready for viewing under a light microscope like we would view in lab. So these are ones you would see in our lab setting. The second one is a TEM. It's a transition electron microscope. Transmission electron microscope, and it is super expensive. This image shows the outside of the cell. They use heavy metal salts to get this gray image of outside. Cell looks pretty neat, right? We'll never use one of these. Super, super expensive. And then this one down here is an SEM. So it's a scanning electron microscope. Again, get a nice 3D image, same as the TEM, except with an SEM, you can see the inside of the cell as well as the outside of the cell. So SEM gives you just a little bit more viewing there. We're going to really focus, like I said, on epithelial and connective tissues in this lecture. Nervous system, again, is going to be later. Muscles and skeleton will be later. They're coming up. And then we'll look at a nerve cell and some muscle cells right there at the very end. But those are also coming up. And that's mostly so that I want you to be able to see the differences, right? I don't want you to we end up going to the muscle lecture and you're like, wait a second, those look like, this connective tissue that we were looking at. So we're definitely going to look at some of those slides at the end, just so you can be able to differentiate the two. Anyway, focus on epithelial and connective. So memorize this slide, make some flashcards over this slide, quiz yourself, but definitely know this slide. You will be asked questions over this, whether it's on the exam, on the quiz, in the lab practical. Memorize the slide, okay? You'll have to know this. And we'll talk about most of these categories individually as well. So we've got simple squamous epithelium, stratified squamous epithelium, simple cuboidal, stratified cuboidal, simple columnar, stratified columnar, pseudostratified columnar, and we have transitional. So you see a lot of these, words repeating: simple, stratified. What do those mean? So simple just means one layer, right? It's one layer of cells. It's one row of cells. Stratified means you have multiple layers or multiple rows of cells. Think tiramisu. Tiramisu is a stratified layer cake. There are multiple layers. It's stratified. Same thing as in cells. Simple, one row of cells, one layer of cells. Stratified, bunch of rows, bunch of layers. So more than one layer is stratified. Stratified could be two layers. Typically when we see stratified epithelium, it's going to be way more than two layers. And you'll see that. You'll see that in a slide coming up. So squamous epithelium, these guys are going to be thin, they're going to be flat, they're going to be scaly. From the top, they look like oddly shaped puzzle pieces. There's not really a lot of space between them. They fit together just right. If we're looking at squamous cells from the side, they look like a fried egg. So they are pretty flat. The only bump that you will see is around the middle of the cell where the nucleus is. Simple squamous, they line the blood vessels and the alveoli of the lungs, just as an example. They line more than just that, but know those two as examples of where simple squamous epithelium lies. Stratified squamous epithelium, they line the mouth, the esophagus, the vagina, things like that. So think about stratified squamous. Stratified means multiple layers of squamous cells, specifically when we're talking about the mouth, the esophagus, and the vagina. Why would you need a bunch of layers of cells in those specific areas? Again, you'll find stratified squamous in more than just these areas, but these are the ones I want you to know for now. So, why would you need multiple layers of cells in these areas? So if you're eating chips, for example, chips can scratch up your mouth, and that's okay because you've got stratified layers of cells, so you can afford to lose some of those cells. If you only had one layer of cells and you were eating something rough or crunchy or, sharp chips or sour candy, you'd be hurting, you'd be bleeding, etc. Now, you can still do that if you're eating something super rough because, your mouth will bleed sometimes. But that just means you've gone through a bunch of layers of cells. If there was just one layer, you would have been hurting from the get-go. So multiple layers of cells just provide more barrier, give you more wiggle room, right? Same with the vagina. This is anatomy class. We're going to be talking about that anatomy too. You'll hear about the reproductive system a lot more. But anyways, vagina, it also has to be able to handle friction. So there have to be multiple layers of cells to line the vagina, whether it's friction from a penis, whether it's friction from a tampon. The vagina's got to have stratified cells to protect it. All right, simple cuboidal. So cuboidal cells, they look like little cubes. Cuboidal, little cubes, little squares. Simple cuboidal line the kidneys or the kidney tubules and the ovary surface. And then stratified, so that's going to be rows of cubes. And stratified cuboidal cells look like a brick wall, essentially. They line the sweat glands and the mammary glands. That's where you can find those. Cuboidal cells, simple cuboidal in the kidney tubules, the ovary surface. These cells absorb and excrete a lot. That's what cuboidal cells function in. Stratified as well, in absorbing and excreting. And then we have simple and stratified columnar cells. So columnar, columns, they're tall, they're skinny, they look like columns. And they're vertical. If it's a vertical picture you're looking at, they're going to look like columns. But if you're looking at a side cut, they're going to be long and skinny, but still they're going to look like columns, and they should be pretty simple to point out. Simple columnar are found in the digestive tract. In the digestive tract, they're not ciliated. So remember, cilia are long, skinny hairs that waft things along. They move things along. In the bronchi and the fallopian tubes, they are ciliated. So the cilia and the bronchi, they get objects that shouldn't be there out of the lungs, right? In the fallopian tubes, the cilia actually waft the egg along its path. So from the ovary through the tubes and to the uterus. In the digestive tract, you'll see goblet cells. Those look like little champagne glasses. And those are going to be full of mucus. In the digestive tract, it's got to be slick to move things along. And that's how food can actually move through the digestive tract. And we'll see some goblet cells there as well. Stratified columnar cells can be found in the pharynx, in the throat. So remember we talked about layers, stratified layers in the lining of the mouth, the esophagus. You also have stratified layers, not of squamous cells, but of columnar cells in the pharynx, which is the throat. Pseudostratified columnar. Okay, so pseudo in science means false, right? Or not really. So pseudostratified, falsely stratified columnar epithelium. So let's talk about it. Okay, that one's kind of odd. You're going to see a picture of it. It's going to make a little bit more sense once you see it. But it looks like it's not really columnar, but it is columnar. So some of the cells look shorter. Some of the cells look like they don't go all the way to the top. They don't stretch all the way to the basement membrane where they're supposed to. Some look like they don't go all the way to the bottom, but they do. They're just oddly shaped. So they can be super thin at one end and thick on the other end, and you can only see the thick side unless you look a little bit more closely to see how it thins out. In the vas deferens, so that's the tubing in the male that moves the sperm from the testes to the penis. It's non-ciliated pseudostratified columnar. And then in the trachea, it is ciliated. And that's the windpipe. So cilia, moving stuff out of the lungs. Trach also has some cilia in it. And then transitional. So on the surface, it looks like it's made of bubbles. Teardrop-shaped bubbles, in fact. Now, transitional epithelium can only be found in the urinary system. In the bladder, the ureters, the urethra. They're going to be in the most stretchy places, right? So the bladder has to stretch when filling up with urine, and then it has to shrink back to normal when urine leaves the body. So the urethra, the tubing in your urinary system, it also has to be able to stretch as urine passes through and then shrink back down when urine gets through. Transitional cells, so they have to be able to expand and shrink, expand, shrink, expand, shrink. So you have to have that extra give. And an easy way to remember transitional epithelium is that they look balloon-like and bubble-shaped. So they can stretch out to be long and skinny and squeeze tight. So they're constantly stretching, constantly shrinking, constantly stretching, constantly shrinking. But now let's look at these guys individually. So simple squamous. So from the side, looks like a fried egg. From the top, it looks like puzzle blobs. No gaps. Makes for a watertight surface. And you can see the nuclei in there. All of those are little simple squamous epithelium. Here, it's of the alveolar air sac walls. Right here, you can see how that looks like a little fried egg. Okay, simple squamous. Simple cuboidal, so this is one layer of cuboidal cells. We'll look at some better size at the end. Don't really love this image, but here is an example of a simple cuboidal kidney tubule right here. So this is one kidney tubule. And then this is a cuboidal cell, 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 and this one can't really see the nucleus in it, but nucleus, cuboidal. Can't really see the nucleus in this one, but this in the middle is the hollow tube. So this is a single layer of cuboidal cells covering this hollow kidney tubule. Basement membrane. The basement membrane is the base layer that separates the epithelial cells from the underlying connective tissue. So what you see between that right here is connective tissue. So basement membrane is the base layer, right? It's gonna be right down there at the bottom. You can also see it called basement membrane, or basal lamina. It's all the same thing. It's the base layer of the epithelial cell. So in this picture, the basement membrane is right there. This picture, the basement membrane is on the outside. Then you got cell, single layer, and then the hollow tubule in the middle. Simple columnar. So all of these cells touch the basement membrane. This part of the cell is called the apical surface. And then this is the hollow middle. So this is a stomach mucosa cell, right? Or this is simple columnar epithelium of the stomach mucosa. So this in the middle, this is hollow. And that's just a simple columnar epithelial layer right there. Basement membrane here, apical surface here, basement membrane here, apical surface here, and a hollow middle tubule. So this is where the food would be, right? If there was food moving through. All the cells are lined up in a row. They're easy to identify. All the nuclei are lined up by the basement membrane. Pseudostratified does not look like this. So notice how all of the nuclei are just lined up nicely together. This is pseudostratified columnar. This is ciliated. So the cilia are right here. These little hair-like structures that are moving things through. This is a pseudostratified ciliated columnar epithelium lining the human trachea. So ciliated, boom, right there. Moving stuff out that shouldn't be in there. Notice how long and skinny and oddly shaped it is. It's like columnar, but the nuclei are not lined up, so they're kind of weeble-wobbly all over the place. Some look squished. Some cells look like they're not touching the basement membrane down here, but they are. They're just oddly shaped, so they're wide up here, and then they get really skinny until they touch the basement membrane. Or they're really wide down here with the nuclei down here, and they get skinny when they touch the apical surface. But they are spreading from the bottom to the top, even though they look very, very odd. If there's a very specific line that cuts through up here, then we would call it, then it would be stratified columnar. But this is pseudostratified columnar. Ciliated, pseudostratified. So not really layered, right? Because they are stretching from the basement to apical surface. But if there was a line here and different layers of columns, then it would be just stratified columnar. Goblet cell. This is a goblet cell right here. So remember I told you it looked a little bit like a champagne glass. So there it is. Boom. Goblet cell. Champagne glass. Stratified squamous. So you can see the multiple layers, extra layers available to fight the friction. Have no idea how many layers these are, but you can see in this one all the different nuclei and how far down that stretches. Basement membranes down here, connective tissue below it, and all of these stratified squamous epithelium. And then we have transitional epithelium, so notice the bubbles at the top, teardrop shaped. These are called umbrella cells, and they got to stretch out when the bladder's full and bubble up when the bladder is empty. You can see that here too. Transitional. So connective tissues have rules. What makes something a connective tissue? So it has to have three things. It has to have ground substance, fibers, and cells. So what is ground substance? Ground substance is the filler material, the stuff that is between the cells. It's what fills in between the fibers, between the cells. It's filler material, ground substance. Number two, you gotta have fiber. So collagen, that's going to be thick and white usually. Now, in our slides, it's going to be stained pink. In real life, it's thick and white. Elastic cartilage is going to be the stretchiest. It's going to look wavy. And usually it's stained yellow or black. And then reticular Fibers, they look like tree branches. They are thick. They are usually stained black or dark brown, and they are like tree branches, sharp and branching. Let's see. And then cells. So connective tissue proper. So cells in connective tissue proper, they're going to be called fibroblasts. They're called chondroblasts in cartilage, osteoblasts in bone, and they are stem cells in blood. So what is blast? When you see blast as a suffix, that denotes immature cells. So we're talking about cells that are immature, they're forming into mature cells. So a mature fibroblast would be called a fibrocyte. A mature chondroblast would be a chondrocyte. A mature osteoblast would be an osteocyte. On that same note, every time you see osteo, we're talking bone. Every time you see chondro, we're talking cartilage. So osteochondro, that's going to come up several more times. So areolar tissues. Some call it loose areolar. It's lacy, looks spacey. These guys are found under the skin in mucous membranes, between muscles, around organs, around nerves. And let's just go through it. Remember the rules? Got 3 rules. So nuclei, you can see the nuclei. They indicate that there are cells here, right? Fibroblasts, nuclei. So check 1, cells. Collagen fibers. So these are going to be the thick pink fibers right there. They're wide. They're elastic fibers. The collagen fibers are wide. They're stained pink. In real life, they're white. And then the elastic fibers are these little skinny guys, and they are stained dark purple. And then everything over here that's really, really light pink, that's our filler material. So we've checked off all 3 connective tissues. Got filler material, got fibers, got cells. And this is areolar. Then we have adipose tissue. Adipose tissue are fat cells. These are going to be the most easy to identify. Each of these bubbles is a fat cell. It's an adipose tissue. But yeah, they look like these giant bubbles. Should have no problem identifying this. You're not going to really see anything else that looks like adipose tissue, or you shouldn't. You can see the nucleus is on the edge of each of these cells. This cell is mostly a vacuole, so almost the entire fat cell is a vacuole. Remember, vacuoles are our storage sacs. Well, makes sense. Fat tissue, storage sacs. So the fat cells are going to be mostly a vacuole. Again, nucleus right there on the edge. There are fibers between these cells, but you can't really see them because this tissue isn't stained very well. And then the ground substance is this light gray material between the cells. So again, check marks all 3. And when you do see this in a lab setting, you're not going to confuse adipose tissue with anything else. All right, reticular tissue. This one should also be pretty easy to identify because notice the tree branching reticular fibers. These guys are found in lymph nodes, liver, kidneys, spleen, tonsils, bone marrow. And there's going to be nothing smooth about reticular connective tissue. The filler substance is going to be gel-like, so that's this white space. And then you can obviously see all of these different cells and then the reticular fibers. So all 3 check marked. All right, so we have dense regular connective tissue here on the left. This is of a tendon. And then we have dense irregular connective tissue here on the right. And that is of the dermis of the skin. So dense regular connective tissue, it's smooth, it's lined up. Collagen fibers are in nice rows. There's nuclei between them. There's white filler material between them. Everything's nice and organized. Same thing in dense irregular connective tissue, except for the organized part, right? So It's going everywhere, made up of the exact same stuff. Got your nuclei, which indicates cells, your collagen fibers, the white ground substance filler material between, but there's no organization here. Hyaline cartilage. Okay, so chondrocytes or chondroblasts. Hyaline cartilage from the trachea is what we're looking at here. And how we differentiate this from anything else, we call them actually double bubbles. So if you see double bubbles like this, we're looking at hyaline cartilage. They also have fibers. Can't really see them here because of the staining. Looks smooth, nondescript. But what we're looking for here are the double bubbles. So double bubbles here, double bubbles here, double bubbles here, double bubble, double bubble, double bubble, double bubble. And that's how you can differentiate hyaline cartilage from everything else. Elastic cartilage. So this is in the aorta, the largest blood vessel in our body. So this has to stretch a lot as blood pumps through. It's got to have recoil, so it's got to be able to stretch, come back, stretch, come back, stretch, come back all day long, nonstop. You don't want it to stop because then you don't have blood flow and then you're dead. So elastic cartilage helps that. Elastic connective tissue in the wall of the aorta. Again, fibers, you can see there. And then connective tissue between, and then if we were able to zoom in a little bit more, you'd be able to see the nuclei. All right, chondrocytes, fibrocartilage. So fibrocartilage of an vertebral disc, special staining produced the blue color seen. So the intervertebral discs, so that's the disc in between the vertebrae and your spine. We see little cells sprinkled throughout. These little cells sprinkled throughout are actually called lacuna. Not laguna, but lacuna with a C and an E at the end. If you compare this, and then obviously a bunch of, collagen fibers. So you can see the fibers, you can see the little cells. And then filler material is going to be in between the fibers. But if you compare this with hyaline cartilage, this is way more fibrous looking. Dense regular also doesn't have these big open cavities where the cells are, so don't confuse it with dense regular. So dense regular has these nuclei sprinkled throughout, and then hyaline has the double bubbles. Fibrocartilage is like a combination of the two, so be able to differentiate fibrocartilage from those other two. So you can see the collagen fiber, you could see the lacuna, so the little cells sprinkled throughout, and then there's filler material there as well. So we're going to look at some epithelial slide examples where you can just get a different look of everything we just talked about. So simple squamous here, just with different staining styles as well. So you weren't just looking at the other side. Simple squamous here, like the puzzles that we were talking about. And here, might look more egg-shaped. So again, pretty easy to identify, single layer. And then we have stratified squamous. So you can see all the multiple layers there. Again, stratified squamous. And they're all touching like puzzle cells. They look a little bit egg yolky, and there's a bunch of layers. And stratified squamous at number 3. So if we look at a slide like this, again, you can see all of the different layers of squamous cells. And we have cuboidal. So again, this is a tubule, right? So that's one single layer of cells. So that's cell, 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 cell. That's a tubule, and that's a hollow middle. So single layer, hollow middle. Another example, single layer, hollow middle, single layer, hollow middle, single layer. Basement membrane there, apical surface there. basement membrane, apical surface. Another image of simple cuboidal. You'll have access to this as well. Oh, this is not a good picture. That one needs to be deleted because that is also simple. Oh, yeah, this is not stratified. That's simple. So let's delete that slide. Okay. Stratified cuboidal here. You can see the layers. Stratified cuboidal here. So cuboidal cell, cuboidal cell, cuboidal, cuboidal, another layer around that, and a hollow tube in the middle. Simple columnar, so you can see how they're stretching from the basement to the apical surface. Another simple columnar. Columnar cell, columnar cell. Those are cilia right up there. So this is ciliated. Then another simple columnar. Apical surface, basement membrane. What I think I said, yeah. Yeah, so apical surface here, basement membrane down there. The nucleus is going to be around the basement membrane and columnar. And like I told you, basement membrane, basal surface, base membrane, all of those are the same thing. Then another simple columnar here. Pseudostratified columnar. So these guys are going to be a little bit less regimented. We got wider columns here, skinnier columns here. Wider, more strangely shaped columns. Really skinny column. Pseudostratified. Same thing here, pseudostratified. You can tell the nucleus isn't lined up perfectly. So that differentiates it from simple. And transitional, so remember we're looking for bubbles. Bubbles. Another one with bubbles up top. Remember, this is in our urinary system, so that they can shrink and expand with a shrinking and expanding bladder, right? Urinary bladder fills up and then empties out. And then some more bubbles. So look for the bubbles in transitional slides. Dense regular connective tissue. Perfect. Perfectly lined up. Got the nuclei, the cartilage, filler substance. Again, perfectly lined up. Look how beautiful. Irregular. So we're taking these two and we're wadding them up essentially. So it's dense irregular connective tissue. Adipose tissue. Again, this one should be the easiest one to identify. They just look like giant bubbles. Well, giant relatively speaking. Elastic connective tissue. Elastic fibers right there. Another example of elastic connective tissue. Curly fibers. And then another example of elastic tissue. Curly little fibers. Hyaline cartilage, double bubbles. Another example of hyaline cartilage, double bubbles. So that's what you're looking for there. Collagen fiber for the wide bands. Notice all of the bands interspersed. Another example of collagen fibers. We won't be looking at a slide like this, unfortunately. This one's pretty cool. And then another example of collagen fibers. So these all look relatively the same as the image we looked in the lecture notes or in the lecture slides. And then we won't be looking at an image like this. It's just pretty neat to see Reticular tissue, tree branches. They don't look like anything else. They look like very harsh tree branches. Cells interspersed throughout. Same thing here. And then we have areolar connective tissue. Again, this looks pretty similar to our lecture notes. Collagen, it's going to be wider. Elastic's going to be a little bit skinnier. And our filler substance in our cells. So it'll look pretty similar. We have fibrocartilage. This is a good one with little notes, arrows. Remember, fibrocartilage, you're looking for the little bubbles between, so you don't confuse it with dense connective tissue. Little bubbles are called lacuna. Another fibrocartilage. So here we're looking a little bit more dense irregular, but again, we have the lacuna, so don't confuse it with dense irregular. This is fibrocartilage. Another example, this is actually a pretty decent example of fibrocartilage. I like this one the most. With the lacuna, the chondrocytes, the little cell bubbles interspersed between the fibers. And another one similar to the one before. Okay, now skeletal muscle. This is what skeletal muscle looks like. Looks different from the rest of it, right? More we'll talk about it more once we get to muscle, but this is what skeletal muscle looks like. Cardiac muscle, smooth muscle, and then this is what a neuron looks like. So this is nerve cell. Again, we'll talk about muscles more when we get to chapter over muscular system, and then we'll talk about nerves more when we get to the chapter over nerve cells. But I did want you to just glance at those. Skeletal muscle, cardiac muscle, smooth muscle, and then a nerve cell, a neuron. And that's it. So look through these. Study these. We'll look at these in lab as well. And that's all.